Hello, I'm Breton, one of our MCAT tutors here at Inspira Advantage, where we help students get into med school and other professional programs. Enzymes are a specialized protein that catalyze chemical reactions in a living organism. They are named based on the substrate they act on and the type of reaction they catalyze. Today, we're going to talk about the common enzyme names and their catalytic properties that you need to know for the MCAT, as well as talk about how enzymes actually work. So let's start off here by saying enzymes can be classified into 6 main categories based on the type of reaction they catalyze. These categories are oxoreductases, transferases, hydrolases, lyases, isomerases, and ligases. You'll note these all end in aces, which will usually tell you that we're talking about an enzyme. Let's take a closer look at each one, starting with oxoreductases. Oxoreductases. These enzymes catalyze redox reactions that involve the transfer of electrons from one molecule to another. Next up, we have transferases. These enzymes move a functional group from one molecule to another. There are 3 types: kinases, phosphorylases, and phosphatases. Kinases add a phosphate group, while phosphorylases introduce a phosphate group into an organic molecule. Typically, this molecule will be glucose. And then finally, for our transferases, we have a phosphatases. These remove a phosphate group. Now we're done with big transferases. Again, to help you memorize these, I strongly recommend throwing these into Anki cards. They are really helpful. A great pre-made Anki deck I would recommend for you is called the Miles Down Anki deck. Just give it a Google and it'll come right on up. Highly recommend for your MCAT prep. Absolutely for use as well. Next up, we've got the hydrolases. These enzymes catalyze cleavage with the addition of water. So this means it's literally using water to break apart a molecule. A common type of hydrolase you're going to run across on the MCAT is called a lipase. And this is a type of enzyme, specifically hydrolase, that's going to break down fats into fatty acids and their glycerol components. Next up, we have our lyases. These enzymes catalyze cleavage without the addition of water or the transfer of electrons. The reverse reaction, synthesis, is often more important biologically, so you're not going to see a lot about lyases, but you still want to know what they are on the off chance they come up. These enzymes catalyze the interconversion of isomers, including both constitutional isomers and stereoisomers. And then finally, we can be done with all the aces. We'll end on ligases. Ligases join 2 large biomolecules, often of the same type. So this is like adding 2 sugars together. If you haven't, I recommend grabbing a screenshot of this and putting these into your Anki cards. It's important to note that some enzymes can belong to more than one category. For example, kinases are a type of transferase that add a phosphate group, while phosphorylases introduce a phosphate group into an organic molecule, such as glucose. Enzymes are proteins that act as biological catalysts, accelerating chemical reactions in living organisms. They achieve this by stabilizing the transition states of the reaction, providing a favorable microenvironment or bonding with the substrate molecules. The active site of an enzyme is the site of catalysis. This is where the substrate is going to bind to the enzyme to form the enzyme-substrate complex. There are 2 main theories to explain how enzymes and substrates interact: the lock and key theory and the induced fit theory. According to the lock and key theory, the enzyme and the substrates are exactly complementary in shape and fit together just like a lock into a key. No funny business, perfect fits between each. The active site of the enzyme is already in the correct conformation to accommodate the substrate. On the other hand, the induced fit theory suggests that the enzyme and the substrate undergo conformational changes to interact fully. The active site of the enzyme is not in the correct conformation to accommodate the substrate until the substrate binds to it, inducing the necessary changes. The induced fit theory is generally accepted as the better and more correct theory, although you should be familiar with both for the MCAT. Now let's talk about molecules that help enzymes perform their functions. Some enzymes require cofactors or coenzymes to function properly. Cofactors are typically metal ions and are required by some enzymes to function. For example, zinc ions are required by the enzyme carboxypeptidase for its catalytic activity. Coenzymes, on the other hand, are organic molecules that are required by some enzymes for functioning. One example of a super common coenzyme I almost guarantee you've heard before is NAD+, which, where is it used? Oh, you guessed it, all over the place. Glycolysis, the ETC, citric acid cycle. This is one of the most important coenzymes for the MCAT, as well as many of your enzymes you're using right now to continue living. Understanding what the names of these enzymes mean will help you problem solve and get more points on test day. It's also really important to know about the mechanism of activity for enzymes, because enzymes are such a high yield topic for the MCAT, spanning both chem phys and biochem sections. So thank you so much for watching our video on enzymes, and I will see you next time.